Thank you, Dan, and thank you so much for this nice invitation. This looks like a very exciting workshop, so I look forward to the interactions and to learn about advances in imaging. I want to talk today about how humans see, and particularly about recent evidence on how we use behavior and eye movements to process visual information. I won't present imaging data, but the visual motor strategies that I will talk about present serious challenges for imaging brain function. A couple of disclosure before I continue. I am co-inventor of a patent on an eye tracker that we use for much, if not most, of our research nowadays. And part of the results that I present today is supported from Meta. Okay, so in all other sensory modalities, it's immediately obvious that behavior plays a critical role in processing information. Right, in olfaction, for example, the sniffing actions determine the strength of the signal itself. So you obviously need to take that into account. Same in touch, even more so in touch, right? The way we move our effectors shape the structures of sensory signals. So we need to process that by incorporating that motor knowledge. In vision, however, vision scientists don't usually think too much about behavior. Of course, we understand that behavior is very important. The vision guides behavior and it helps selecting information. But the idea on how space is represented in the visual system starts from the fact that we have an image that is passively formed on the retina. And so space is primarily encoded by the locations of neurons within maps. That's the standard idea on which most theories of vision science rely. And this has led to several theories that are essentially formulated as image processing techniques. We are passively processing images. For example, how is a scene represented, right? The idea is that the brain processes information through a hierarchy of more and more complicated filters that extract useful information. For example, they can extract edges, and then we achieve the representation of the world, how we perceive it. Or take stereopsis. We mentioned before the cue of stereopsis as a disparity, as a major cue for depth perception. We perceive depth also because we have two eyes, right? And we use this cue. The idea on how we use it is that we start from the two images. There are slightly differences in the two images, and then we try to find those images and establish corresponding points. Or even developmental theories of disorders, right? Most of them rely on thinking about passive static images. For example, myopia, which is endemic nowadays. How is myopia developing? How is the eye not emmetropizing, not finding the right distance, the right length during development? All theories that are out there are based on the quality of the image on the retina. Now, we know that these ideas are important. We have learned a lot from them, but we also know that we are missing out one component, because we know that neurons are not just spatial filters, they're also temporal filters. They have their own dynamics, they respond very strongly to transients. And we know that there are no images on the retina per se, right? There is a spatiotemporal flow that enters the retina because we are moving all the time. And that leads me to the main point that I want to make today, which is that spatiotemporal flow, those temporal transients that are impinging onto the retina, mostly come from us, from our own behavior. So to convince you of that, let me take you to my hometown. That's a landscape of Tuscany, just outside Florence. And as you're looking around this scene, you're probably not paying too much attention to your eye movements. But what you do, you shift your gaze from one point to the next by means of rapid gaze shifts that are known as saccades. This is probably the most frequent movement we make, more frequent than heartbeats. And we spend just a few hundred milliseconds in each fixation point in between those saccades, so that the input to the retina looks something like this. Now, you may notice that even in the period of fixation, even in the periods in between saccades, the image continues to move, and that's something that I will address quite a bit later on. So imagine a receptor sitting on the retina, here it is, And now this receptor will be continually exposed to changes in its input, to changes in luminance. And this has traditionally been regarded as a major problem in vision science. How can we establish a stable 3D representations despite all this? There have been innumerable papers on various questions related to this point, always framed in negative terms. Why is the world not moving for us? How can we achieve a stable representation? How come we don't perceive the big motion caused by saccades? And why is vision not fragmented by the different fixations? Or even during one single fixation, given that the eye is moving all the time, as you've seen from that moving, how come the image is not blurred by that movement, given the temporal sloppiness of neurons in the retina? Now, this talk today focuses on an alternative approach that our laboratory, others laboratories have been following for the last 2 decades, which reverse the problem and regard this as an efficient strategy for establishing spatial representations. Now, the essence of the idea is the following. Let's go to a 2-dimensional world, a flat land, and let's look outside so that the input to the retina will look something like this, right? Now, if your eye doesn't move, so let's acquire information statically, each receptor is sampling from a different point in the scene. And if we don't move, we have no other way to learn about space than to integrate information across many receptors. So space is represented by the topographical representations of activity in neuronal maps, the standard view in vision science. But let's move our retina, let's move our eyes. So what we are doing now, we are introducing temporal changes, and that temporal transience conveys spatial information. Now, if I want to decode, if I want to extract spatial information, I have a variety of techniques, a variety of strategies that I can follow in the joint space-time domain, right? In the very limit, even just one single receptor tells me about how space is organized in a neighborhood by means of its own changes. So we call this idea active space-time encoding because it's mediated by behavior and is in the joint space-time domain. And here are a couple of reviews if you're interested in learning about this. It really doesn't take much to convince people that what we see depends on how we move. And I hope that, this is a very good projector, so this should work. So if you fixate on this image, fixate means maintain your gaze steady on that red dot, you may just see what vision scientists call a grating, right? A high frequency sequence of black and white stripes. But if you now move your gaze following the red dot, you may realize that there is another image there. Is this working? It depends on the distance which you are. Okay, I see a few people nodding. So you may see that there is a face underneath that grating, right? If you now start following it. Okay, so what's going on here? Well, actually, in this picture, there were 2 images. One was at higher spatial frequency, sharper variations, and another one at lower spatial frequencies. But the eye moves. So what happens? As the eye moves, these images are transformed into temporal changes. They are transformed into temporal frequencies. Here they are. And the way they are transformed depends on their structure. So the finer details, the final details get transformed into faster variations, higher temporal frequencies. Now, the visual system is not equally sensitive to all these temporal changes. In fact, there is a range of temporal frequencies, of temporal rate of change that the visual system really prefers. And by moving in different way, we can bring one of these 2 different images within that range of sensitivity. That's why we unmask the grating and we now see the image that was at lower spatial frequencies. So the idea is that eye movements always do something like that. But the way they do it depends on their specific dynamics. And different types of eye movements will transform that spatial pattern into different types of temporal modulations. None of them will move the image at uniform velocity on the retina, but they will continually change velocity. So what they will do, They will spread the signal of the image out there into various rate of temporal changes, various temporal frequencies on the retina. Okay, so let's look a little bit at how different types of movements reformat the input to the retina. And to do that, I go back to this slide in which you have seen, you see that there are the various points of fixation highlighted, and then there are saccades that move from one fixation point to the next. And let's focus on one fixation, okay? So if you have a very good eye tracker and you can follow gaze quite precisely there, you realize that your eyes are also moving during those periods. You have seen an example in the movie that I showed before. They continue to move primarily by means of two types of movement. First, you have, again, small saccade. They are replicas of the larger gaze shifts that we perform, but they are much smaller. And historically, they got labeled with a different name. They're called microsaccades. They maintain the image within the fovea. The fovea is the high resolution part of the retina, which is just 1 degree at arm length, your fingernail at arm length, 1 degree. In the periods in between saccades and microsaccades, an incessant jitter of the eye continues to move the image across many photoreceptors. We call it fixational drift, ocular drift, or eye jitter. You can see a trace here. And here is an example of that eye jitter in your own eyes. We are not aware of doing that, but what you're looking at here is a static image, and the jitter of your eyes combined with the jitter of your head is creating motion signals that now become apparent to you. And so that's why the object appears to move. What you're experiencing now is the motion generated by your own eye movements. So these are large movements relative to the size of photoreceptors and relative to the size of the fovea. Now, the standard idea, we have known that these movements exist for a long, long time. And the standard idea is that these movements are there because they are needed to refresh neural activity. For example, here you can see there is a phase in this image, is a faint phase. And if you fixate, if you maintain your gaze steady on that red cross, the image will fade away. We call this phenomenon Troxler fading. So those eye movements seem to be necessary to prevent this from happening in reality, in the real world. Well, what I hope I'll convince you is that they do much more than that. They're not just refreshing neural activity, they're structuring neural activity. They're providing transients rich in visual information, in spatial information that is then decoded by the visual system. So let's think about that a little bit, and let's focus on the case of eye drift, which is easier to start from. Okay, here is a photoreceptor, the red circle, looking at one particular grating, these black and white stripes, and as it jitters because of eye drift, it's experiencing temporal modulations. Of course, these temporal modulations will depend on the stimulus itself, right? Let's consider the same motion on a lower spatial frequency stimulus. Here it is. In this case, we don't get much of a change. And if we look at a higher spatial frequency stimulus, we may get similar amount of change as before, but now the rate of change is much faster. It's going to be much higher temporal frequencies. Now, within the range of temporal sensitivity of retinal neurons, these modulations have different effects. The intermediate frequencies will provide very strong modulations, for example, versus the lower frequency, the lower spatial frequency. Do I have a pointer here by any chance? Do you see my pointer? No. You know if I have a pointer? With the mouse, I see on my mouse here, but not on the monitor. Yeah. Oh, okay. Okay, thank you. Okay, so let's assume this is the peak sensitivity, close to the peak. This means that the modulations are strongest in the temporal frequency range of sensitivity of the retina. And of course, a lower spatial frequency stimulus only provides less signals, less power, because you don't get much of a temporal change, versus these signals will provide changes that go out of the temporal range of sensitivity of retinal neurons. So you see that because of the temporal sensitivity of neurons, the space-time conversion operated by the space-time reformatting operated by eye movements acts as a sort of spatial filter, right? But is only in the temporal domain. There are a couple of interesting things about this spatial filter. The first one is that it seems to process information efficiently. If you look at how the strength of these modulations increases, there is a broad range in which they increase proportionally to the spatial frequency of the stimulus. Now, we live in a world that has exactly the opposite structure, the inverse structure of this, in a world that is dominated by low spatial frequencies. And the power of natural images tend to decline proportionally to spatial frequency. Now, these two effects counteract each other. What that means is that the jitter of the eye creates modulations that are equal in amplitude during different spatial frequencies, during viewing of natural scenes. You can look at these as a sort of matching between the luminous modulations from the eye jitter and the natural visual world. It's interesting because this equalization or whitening, as is usually known, is a standard technique in signal processing to improve the efficiency of transmissions and representations. Now, there is something else that is interesting about this, which is that potentially, if we are in control of our eye movements, in particular the drift in this case, we could flexibly tune this filter via motor control, right? Let's think about that 1 second. So here, let's assume that this signal, this image, is the one that gives the biggest modulation. So it's this spatial frequency here. Now, with this amount of motion, if I enlarge my eye jitter, but I also stretch the stimulus, I can produce, again, the same modulations. What that means is that by moving more, I'm shifting that filter toward lower spatial frequencies. Or by moving less, I can filter that filter toward higher spatial frequencies. So the amount of motion will determine the range of frequencies that I'll be responding most strongly to. I can select information effectively in this way. But let's ask ourselves the main questions. Are these modulations really important? Now, to understand that, we need to have very good eye trackers because these are small movements. They're small for the experimenters. They are large for the retina. And most of the data that I will be presenting today are data collected with a dual Purkinje image eye tracker. This is an eye tracker that compares reflection of an infrared beam from the cornea and the back of the lens and can go down to arc-minute resolution. And again, arc-minute will mean 1/60th of your fingernail at arm length. Now, we have re-implemented this system in digital form, and that's what we use nowadays for most of our studies. But the nice thing is that this system is integrated in our laboratory with another system for gaze contingent display control. This is a system that allows us in real time to modify the stimulus with a certain delay, guaranteed to be a certain delay, so that we can perturb that visual motor loop and understand how sensory information and the acquisition of sensory information evolves with these movements. So let me give you a practical example. Normally, when we run experiments in vision science, the stimulus for us is what is on the monitor. But we have no control of what is happening in the retina of the subject because different subjects will move in different ways. With an apparatus like this, we can gain control of that. We can, for example, move the stimulus together with the eye, right? We stabilize the image on the retina, retinal stabilization, or we can superimpose on that the motion that we want to achieve a level of controlled motion on the retina and really study how that motion affects perception. Now, I would argue this is a major step forward relative to the old techniques of image stabilization, retinal stabilization. This is one of the first. It's a huge, it's called the suction cup. It's a huge contact lens with a pump. You can see here a pump to make vacuum so that it sticks. Then it has a pole and a mirror on top. So you can flash an image here on that mirror, and that pole will move with the eye. And so the image remains stabilized. Our subjects will not be happy to go through this. Okay, so what comes out from these experiments? The first thing the main thing that comes out is that the jitter of the eye enhances perception of fine visual details. So for example, if we ask subjects to tell us whether a fine grating, it's a noisy grating, is tilted to the left or to the right, like here, and we adjust the level of noise so that it's not too easy, but they can still do it. It's challenging, right? They get entertained during normal viewing. If we stabilize the image on the retina, so we remove the temporal modulations, then they can no longer do it, as if those temporal modulations are important for performing these tasks. And this is specific for high spatial frequency stimuli. If I present low spatial frequency stimuli, and now those temporal changes are not that prominent anymore because it's a more smoothly varying stimulus, then performance don't change under retinal stabilization. In fact, these luminous modulations really seem to drive sensitivity. And we can test this by controlling precisely the amount of motion rather than just looking at things normally, like in this case with the normal motion on the retina or stabilizing the image. We can reduce the amount of motion, or we can increase the amount of motion by moving the stimulus in the opposite direction during the jitter of the eye. Okay, so if we do that, and we measure, so we have a variety of motion on the retina, and we measure the sensitivity of individual subjects, so we measure their threshold. Well, there will be a range of motion that is optimal for seeing, and smaller and more motion in an asymmetric manner brings performance down. Now, what is interesting about this is that if you just take the temporal modulations that are produced on the retina by this procedure, and you look at how much signal there is within the temporal range of retinal ganglion cells, parvocellular neurons in this case, that are likely to respond to this type of stimuli, then you see that there is a very close match. This is not a model. You're just taking parameters from the neurophysiological literature and just comparing the strength of the signal within the temporal frequency to psychophysical data. So there is a very good match between the two things. I mentioned before that in principle, we could tune that filter coming from the eye jitter via motor control. And in particular, I mentioned the fact that if we reduce the amount of motion, we can push that filter toward higher spatial frequencies, okay, like this. So we get a higher range of spatial frequencies. But is ocular drift controlled? By now, we have quite a lot of evidence that ocular drift, movements at these scales, are actually controlled. One of the best examples is something we do, all of us have undergone through, and is looking at a 20/20 line on an eye chart, on a standard Snellen eye test. When you do that, it's a fabulous example of control of eye movements, because what you do, you make very small saccades, microsaccades, from one optotype to the next. These are just 10 minutes of arc saccades. They are smaller than the saccade that you make if you're asked not to move at all. If I give you a dot and you're asked to maintain fixation on a dot, these microsaccades that occur there are larger than this. And the jitter that occurs on each optotype is also changing its characteristic. It's different than the jitter that you perform when, for example, fixating on a dot. In fact, compared to that kind of jitter, fixation on a single dot, just maintain your eyes steady. The jitter that we perform on each optotype in a Snellen eye chart is going to be slower and more curved. And the combination of these two together is pushing that filter, that the amplitude of the temporal modulations, toward higher spatial frequencies, so that we are highlighting the range that is really relevant for this task. We are producing a more effectively contrasted image on the retina, right? But in the temporal domain, in those temporal modulations. Interestingly, we can evaluate how much eye movements matter in this kind of test by stabilizing the 20/20 line on the retina. So this would be the combination of both microsaccades and drift. But when you do that, performance drops down dramatically. You really need to move your eyes. But we can recover performance. We just need to enlarge the stimulus. So if we make it much larger, then we go back to normal. That impairment is 0.15 logmar, which is the equivalent of 2 lines on an eye chart. So moving well your eyes is buying you 2 lines on a standard Snellen test, right? Now, it seems that different individuals have different capabilities in terms of controlling their drift. And here is an example. These are all emmetropes. These are all subjects that are capable to read perfectly without correction a 20/20 line. But there is individual variability, so they vary in their own actual acuity threshold. And they also vary in their eye movements. And the two things are strongly correlated. Those that move less, that have smaller jitter, they all have jitter. If you stabilize the image, they won't perform. Those that have smaller jitter are the one with the highest acuity, and vice versa. I just give you another example of drift control, because this doesn't happen only with high acuity tasks. Apparently we tune it according to every task, according to the information that we want to grasp. And this is evidence coming from another type of eye tracker, a big coil system in which subjects wear eye coils, but it's intrusive, but now they are finally free to move their head around, and we can record their movements at that same level of precision that I showed you before, while the head is free to move. And so now they can perform real tasks in the 3D world, like, for example, pointing at objects in the scene, performing a Snellen eye chart while moving their head, or grasping objects and sorting them according to their color or to their shape. Different tasks lead to different speed of the image on the retina, irrespective of how much the head is moving. So we are really trying to tune that amount of motion according to the task that we are performing. And the evidence is that it's according to the information, the scale of the information that is relevant to that particular task. Okay, enough about jitter or eye drift. Let's talk a little bit about larger eye movements, and in particular, saccades, right? That are the main movements that we perform all the time. Is this strategy of encoding spatial information in time a general strategy that applies also to other types of eye movements? Well, saccades will be the movements that provide the most prominent transients. Now, saccades are very highly structured. Here is an example of their main sequence, the relation between velocity, duration, and amplitude. They have very specific structure. Another way to say this is that if I take a saccade, just a small saccade, here is the displacement of the eye, and I scale it in space and time, I can replicate every other saccadic waveform. So they have very, very specific dynamics. They start from velocity 0, of course, and they end with velocity 0 with a very given dynamics. What that means is they're producing very specific temporal transients on the retina. So what are these? Well, let's go back to our filter implemented by the jitter of the eye. And I remind you that it depends which jitter, because if you look at the Snellen eye chart, you push toward higher spatial frequencies. What do saccades do? Well, saccades are going to enhance lower range of spatial frequencies. So there is a range in which the amplitude of the modulation seems to be constant. And then there is another range in which instead the amplitude of the modulations increases with spatial frequency again, like for drift. So there is again a range of equalization, right? So some sort of tuning or matching with respect to the natural world. If we make larger saccade, we push the transition between these two zones toward lower spatial frequencies. And if we make smaller saccades, we push it closer toward higher spatial frequencies and closer to the range of ocular drift. So there is really a continuum, there is really a continuum in the temporal modulations that saccades and drift produce, even though these movements appear so different from each other. We can trace these transients. We can see these two regimes of whitening and saturation even in the cortex by looking at waves that are evoked by saccades. So every time we perform a saccades, we're going to have lambda waves, they are so-called. And the peak of those lambda waves with spatial frequency follow the structure that I showed you before. So they reach the cortex, these modulations. But what do they do? Well, let's see what an ideal observer would do with this observation, with this information. Let's say that I want to extract information at high spatial frequency, a range in which saccades and drift are producing both useful information. Well, the best thing I can possibly do as an ideal observer is to keep integrating that information after a saccade. First, I take the information from saccade, and then with prolonged exposure, this will be the time of exposure to the stimulus after the saccade. With the eye jitter, I keep increasing, I keep doing better. But if the stimulus is at lower spatial frequency, then in this range, ocular drift doesn't provide much information. So in that case, I better just listen to what the saccade is doing, and I won't improve much my perception during fixation. That's what an ideal observer would do. And that's interestingly, is what humans actually do. These are data measured by from human subjects. And this improvement during post-saccadic exposure, the time of exposure to the stimulus after a saccade, that you see for a high spatial frequency stimulus is caused by drift, is caused by the eye jitter. Because if we eliminate it by retinal stabilization, as I showed you before, performance drops down. And this initial improvement in sensitivity after a saccade, that comes from the saccade transient. So if we remove that transient, and we have ways to do that, then performance again drops down. So not just drift, but also saccade seems to contribute useful transients to perception. Now, in vision science, the standard idea is that we don't acquire information during saccades because we don't see the shifts, right? Somehow vision should be suppressed. But the fact that our visual awareness is suppressed doesn't mean that we are not acquiring and using information for establishing spatial representations. And a number of studies have now shown that that seems to be the case. Not only saccades, but also other types of eye movements seem to be benefiting from, seems to produce temporal transients that are used by the visual system. When we track objects moving on in the world, we use a type of eye movement known as smooth pursuit. But the smooth pursuit is not perfect. It will move at different gains relative to the object. The object will change speed in the world. And sometimes we'll have to perform saccades to compensate for that. So there is going to be quite a bit of motion on the retina, as you can see here. In fact, the motion that you experience during smooth pursuit is similar to the motion that you experience when you maintain fixation on a dot and try not to move your gaze at all. And if we ask subjects to perform fine discriminations during smooth pursuit, and each dot here is one subject, well, and we look at their temporal signals, the subjects will have different level of performance. Some of them are better at doing this task than others, but their performance depends on the strength, is correlated with the strength of the temporal modulations that the motion of the stimulus on the retina is producing. Now, I think I have a little bit more time, and so I like to expand a little bit. So far, I've been telling you about how eye movements contribute temporal transients and how these transients help with visibility, with sensitivity, right? Primarily contrast sensitivity. But is this encoding of space in time a more general strategy that the visual system is using? Does it use to extract other features as well, in addition to just changes of luminance? And to investigate this, I'd like to focus on depth perception. As Dan mentioned, depth perception in humans is carried out by a variety of cues. We use around 20 cues or so to establish, and we integrate them to establish a three-dimensional representation of the scene. But a very important cue is stereopsis, right? Are the differences in the images from the two eyes. Those are what we call binocular disparities that emerge from having a displacement between the two eyes. And now, by finding those correspondences, we can triangulate and establish where objects are in the world. That's a standard textbook idea. Now, this textbook idea, it usually neglects the fact that the eyes are moving. They're moving all the time, and they're not moving exactly equally to each other. In fact, there is quite a lot of discrepancy between the two eyes, as I'll show you in a second. And so what that means is that the disparity signal actually is changing all the time. So if you were to look at we call this the doors of heaven. This is the doors of the main church in Florence. And if you're looking at them from 1 meter distance with a standard variability of the two eyes, this is the range of disparities that you will be experiencing. So there is, relative to the capability of the human visual system, these are huge changes in disparity. You can go below 1 arc minute disparity and be able to establish representations based on that. So how are the two eyes moving together? Or how are they not moving together? Well, so here is fixation. The subject is trying to maintain fixation on a dot with the two eyes. You can see red and blue are left and right eyes. And you can see that whenever there is a rapid movement, a microsaccade in this case, the two eyes move together. They are quite strongly correlated. In between microsaccades, they are less correlated. In fact, there is some level of correlation, but it's very partial, okay? And it depends on the task also. Typically, with the head immobilized, very, very low correlation. This is right eye velocity, left eye velocity. Okay, so we can study what the temporal modulations in disparity, the cue for depth perceptions that we are studying here, what those disparity modulations do for establishing spatial representations by using the same technique that I mentioned before. Rather than allowing subjects to look at stimuli with the two eyes normally, we can stabilize the images in the two eyes, right? And we can see what happens then. So here is an example of how this kind of experiments work. The subject is looking through a stereoscope at the two stimuli. And in this case, to show you, we are only stabilizing one eye, not the other one. So you can see that one eye is jittering. Again, these are large movements, right? Relative to the retina. They're difficult to record, but they are large relative to photoreceptors. Okay, so here is the task of the subject. We present them a corrugation in space, and they have to tell us whether that corrugation is tilted to the left or to the right. The same experiment as I showed you before with just grating in 2 dimensions. Now is a 3-dimensional stimulus. We do it with random dots. These are patterns of dots that have been displaced, so to recreate that corrugation. And you can control the level of noise by determining how many of those dots are not correlated in the 2 images. Now, we are very good at these tasks, and subjects can do it very easily when they are allowed to move their eyes normally. But if we stabilize the images, then performance drops drastically, as if these modulations are important, right? So stereopsis is impaired under retinal stabilization. And it doesn't depend on microsaccades. It's really the jitter of the 2 eyes that matters. Only if I isolate trials in which the eye jitters without microsaccades, that effect is very strongly there. But the question is this a monocular effect? So certainly eye movements seem to be beneficial for stereoscopic judgment. But what I'm seeing here is this the consequence of the reduced sensitivity in each eye that I explained before? Is this what is happening here? Or is really telling me something more about a general strategy for now encoding disparity changes rather than luminance changes? So to answer that question, I want to present the data from 2 different experiments. Here we are stabilizing different components of eye movements. I showed you before a slide in which, here it is, in which I show you the right velocity of the eye and the left velocity of the eye, right? I can decompose that motion I can change my coordinate system and decompose that motion as the joint motion of the 2 eyes and the opposite motion of the 2 eyes, version and vergence. It's just a different frame of reference, which makes particular sense when you're looking about binocular eye movements. So how are the 2 eyes moving together and how are they moving in the opposite direction? Okay, so what we are doing here, we are stabilizing those components of eye movements. So for example, we are allowing the eyes only to move, to experience changes caused by vergence or only changes caused by version. And in this specific case, performance drop when we eliminate vergence, when we only allow version. This is an experiment in which only vergence movements provide disparity modulations. So they do well in these experiments with vergence, but they're also experiencing disparity caused by vergence. It happens because the stimulus is designed so that only vergence movements provide disparity modulation. Here is another experiment, different stimulus, designed so that both vergence and version provide changes in disparity. And now performance is normal under normal viewing, impaired under stabilized. If we allow vergence or version to occur, performance recovers. Okay, whenever there are changes in disparity, then performance seems to do better. So this really suggests to us that it is the change in disparity which is critically important for performing this task rather than absolute disparity per se. I think I'm pretty much out of time, so I'm going to summarize. I've spoken about how eye movements continually modulate luminance signals on the retina, and they reformat the input to the retina in a spatiotemporal flow. I mentioned that they act as a sort of filters, but implemented in the joint space-time domain. So they create modulations that enhance different ranges of spatial frequencies depending on the characteristics of the movement themselves. Different movements form a continuum in their modulation. So they are sort of a repertoire from which we can choose to process information in different ways, pre-process information in different ways, according to the tasks. All of these movements exhibit a range in which they seem to be tuned to the natural world. And I mentioned this body of evidence showing that oculomotor modulations go beyond these. They are part of a general strategies for encoding space in time. And thanks to all the people who really helped me with the deep experiment, I mentioned, I put the faces of several of them during the talk, Gianni Sinto in particular, and Wei Wu, the person who is behind the design of our eye tracker, our collaborator, Jonathan Victor, and of course, the sponsors. Thank you.
Well, thank you so much for introducing us to the dynamics of vision here, literally the dynamics. I hope people are thinking about what resonance this might have for some of our artificial imaging techniques. Any questions for Dr. Rucci? Please. Yeah, Ricardo, go ahead, William.
Two short questions, if I might. First of all, the structure of the eye is clearly discrete. Even in the fovea where you have very densely packed cells, there's separate little sensors. And there's effective jittering of the positions actually sort of by moving the image over these effectively allows us to achieve some super resolution in a straightforward sort of way, I think.
Yeah, so in fact, there are various theories about how this may achieve. And one idea is that perhaps this is similar to the super resolution algorithm in computer vision, right? You can have a camera acquire a higher resolution image by moving and taking different images. So perhaps you're doing something like that. What seems to come out from our experiment is something like that, but not spatially, is the temporal changes that are really relevant. So the dynamics of how we move is really relevant, rather than the standard, yeah.
The other question, when we're changing our direction of vision, we're also changing not only the direction of space, but also position of the eye. And so they're actually introducing some spatial discrepancy depending on depth, even with monocular vision. So in response to Dan's comment and your comments, that it actually provides some discrepancy information. It's lower than the separation between the eyes, but there's still some information in there, I would think.
Yeah, that's a very good point, and additional information there to be extracted for the visual system. Absolutely, yeah.
Quick question. Thank you. Amazing talk. So about 10 years ago, I read this article. They were saying that if a kid has strabism, I say the cross eyes, and you don't correct it before the age of 2 or the age of 3, they will never have 3D vision. But is this still valid? How do you explain it? Because they still can move the eyes, right? It's just they're not.
Yes, but that's a different phenomenon. So what happens is that if the correspondence of the two images is not established early, then it becomes really difficult to do it, and vision from one eye gets suppressed most of the time. And so, yes, the eyes continue to move no matter what, but I don't think it's contributing to that. It's something different, yes.
Okay, thank you.
Thank you. That was a terrific talk. I have a question about the potential evolutionary advantage of a system like the one that you described. If we intend to capture something in space that is in motion, apprehend, I don't know, something that's moving through a sphere at a running buffalo or something, or if I were a frog trying to catch a fly moving in space, is our ability to predict the future position in space of a moving object enhanced through these very rapid, repeated views that we're producing through this jittering and almost like an optical flow sense, where I could say this buffalo is going to be 1 foot further and back to the left, and I'm going to time the throwing of my spear. Do you derive any insights like that about the future position of something?
Yeah, it's a difficult question. I don't know if the motion of the eye would allow you to do better prediction of motion of external objects, right? It seems to add a confound, but so I wouldn't know how to answer to that. I think it's not probably helping too much in that sense to establish prediction, but it's probably using similar strategies to this, not even not hurting the process. So I don't have a clear answer to that. What I can think in terms of evolutionary terms is that, so what we think about eye movements is that they evolved to stabilize the image on the retina. So something moves, you want to follow, so you start moving your gaze. But as that process was evolving, and as neurons are very sensitive to times, and also they were evolving, their sensitivity were evolving, I think at some point there was no advantage to go any farther in stabilizing the image on the retina. And so that's why this kind of motion and this kind of strategies evolved. Yeah.
Thank you.
Okay, thanks. So I have two questions. One is that
Can you speak a bit louder, please?
yeah, so I have two questions. One is that this is innate for this eye movement, means that it's not a learned, it's just naturally occur, because, you know, I just have a recent opportunity to look at the infants. I look at her eyes, seems like they move less. That's my one question. The other question is that, you know, we have this, I heard this, we have some dwell time for eyes, the sensitive to dwell time for acquire signal from the outer world. So I see your, the eye movement is very rapid. I don't know what's the unit of arc minute per second. So, okay.
So I'm not sure I heard completely the second question. I'll start for the first. We know very little on how, well, people learn to do different tasks and then they start to move differently in those tasks. There is no doubt. But we have no idea of how much of this is innate. So recording at this level of resolution have primarily been done with adults. So very little experiments with children, for example, and how much their eyes are jittering. Is this level of motion contributing in any way to ametropization, for example? Is it contributing to myopia? We have no idea. And I'm not sure I heard your second question, I'm sorry.
Oh, so for visual processing, we have some kind of dwell time, right? For a signal, you can actually register to the neuron and then react to it. I thought there's something like a 100 millisecond.
Yeah, so retinal ganglion cells will integrate over a scale from 50 to 100 milliseconds and more, depending on, there is a huge variety of neurons. So the standard view, if you want, is that they're a little bit sloppy because they are responding with this dynamics that is there. But if you look at how they are changing the response as the stimulus is changing, then you can get much more precise in time. And I'd be happy to talk more about that.
Yeah, thank you. It's very thought-provoking. Thanks.
Yeah.
Thank you for a very nice talk. It seemed like a lot of your talk focused on kind of the interactions between foveal vision and the temporal domain. And I was curious because, of course, the retina encodes multiple kind of levels of acuity or spatial frequencies across it, and where in the periphery we're encoding much lower frequencies in space. Is there work that is actually trying to understand the interactions between the spatial encoding of the retina and the temporal encoding that you were talking about?
Oh yeah, absolutely. Across the retina, across various eccentricity. Yes. Lots of ongoing work. And interestingly, even though these movements are, so the receptive fields, the region of space from which neurons respond to increases as we move out with eccentricity. So only the fovea resolves very high acuity, right? But so one may think that as we go in the periphery, this motion is going to be too small to activate those neurons because it's going to be just a small change. If we stabilize images in the periphery, performance drop even more than in the fovea. So I think what is going on is perhaps the temporal compensation is, there is more sensitivity to temporal transients. There is quite a bit of evidence for that. That depends who you talk to. But the fact is that retinal stabilization or altered motion in the periphery is as effective as a cue for disrupting things as in the fovea itself. Yeah.
That's really interesting. Thank you.
Yeah.
One last question.
Hi, I was wondering the exploration through saccades of the whole scene, is it in some way predictable or given some features, and which are those features in case?
Yeah, lots of work in that area, right? People are trying to understand what is salient about different parts of the image and what's going to attract saccades. Can you predict saccades? Lots of work also in virtual reality in that context, right? We would help to improve displays. So it's partly predictable. You can have models of saliency that do a pretty good job in determining which would be the landing points of saccades. So we'll highlight a number of points. There is a huge variability across individuals. So it's really, really difficult to predict what one subject will do at any given moment in time.
But there's no features in the scene that is able to give some, the textural or the intensities doesn't give.
Well, I mean, everything that is salient will attract the movements. The best way to, the most systematic thing I know of is actually how to suppress a saccade, which is every sudden change will suppress movements for a while. Yeah, including microsaccades.
All right, let's thank Dr. Rucci again.
Thank you.